The last structure we're going to look at is the quaternary protein structure, and this occurs when several polypeptides bind to form a larger ordered structure. So the polypeptides are held together by the same interactions that we looked at for the tertiary structure. Also, with quaternary protein structures, it can contain non-amino acid portions that we're going to actually look in this example, hemoglobin. So hemoglobin is an example of a quaternary protein structure, and it's made up of 4 polypeptide chains. So you can see that in the picture with the different colors. So the green, blue, yellow, purple are 4 different polypeptide chains. And then within those, there are 4 heme groups. So here's 1, 2, 3, and 4. So each of those peptide chains has a heme group that the oxygen can bind to. And just like what we saw in myoglobin, hemoglobin transfers that oxygen throughout the body. Another example of a quaternary protein structure is collagen. So collagen is the most abundant protein in the body, and the structure is made up of 3 alpha helices that are woven together, kind of like a braid, to form a triple helix. And then the chains, those chains are held together by hydrogen bonding. So you can see in the picture, we have the yellow, the blue, and the purple, different polypeptide chains woven together and held together by those hydrogen bonds. So peptide bonds can be hydrolyzed. So we've been looking at hydrolysis throughout each of the functional groups. So peptide bonds can also be hydrolyzed, and it basically breaks the amino acids apart. So it can give us individual amino acids. So this process occurs in the stomach when enzymes like pepsin or trypsin catalyze the hydrolysis of proteins to give amino acids. So hydrolysis will disrupt the primary structure. Okay, so here's an example. We have amino acids here, and then the peptide bond is broken where it was formed. So right here, the peptide could be broken here and here to give us the individual amino acids, alanine, glycine, cysteine, and aspartate, or aspartic acid. So we do have enzymes that will hydrolyze or break the peptide bonds at specific points within the sequences. So these are called endoproteases. So 2 of them are listed here. We have chymotrypsin, and it will hydrolyze a peptide bond on the right-hand side, or the C-terminus side of amino acids with aromatic side chains. And then trypsin hydrolyzes peptide bonds on the right side, the C-terminus side of lysine and arginine. So just to give you an example here, if we were to take this peptide that's listed and use chymotrypsin, then it would be hydrolyzed here next to tyrosine, because tyrosine has an aromatic ring, as well as right here next to phenylalanine. So that's where the peptide bonds would be broken. Now, if we were to use trypsin, then the bond would be hydrolyzed here with lysine. So that's just an example of where we can, enzymes that can break those bonds at specific points in the primary protein structure. Okay, last of all, a protein can be denatured, and denaturation of a protein occurs when there's a disruption of the interaction between the R groups. So we know with the tertiary structure and the quaternary structure, as well as secondary structure, they're held together, with those interactions. Secondary, it's really just hydrogen bonding between the peptide backbone But tertiary and quaternary structures are held together with those interactions between the R groups of the amino acids. But once those are disrupted, it removes or it loses that structure. So you can see here in the picture, here is our tertiary structure, and then it's denatured, and now we have our primary structure. So the primary structure is not affected, it stays intact, but the secondary, tertiary, and quaternary structure will be disrupted. Okay, and here are the agents that can cause denaturation. So the first one is heat. So heat can denature proteins by breaking hydrogen bonds, and they disrupt hydrophobic interactions that we talked about. Mechanical agitation. So the whipping action, what it does is it stretches the polypeptide chain until stabilizing interactions are disrupted. So an example of that listed here is beating egg whites. So if you've ever made like a meringue and you put egg whites into a bowl and then you beat them until it gives you like this white fluffiness. So that's kind of an example of that. Okay, detergents. So low concentration of detergents can disrupt the hydrophobic side chains. It disrupts those water-free packets that they form, water-free pockets that they form. Organic compounds will disrupt the hydrogen bonding because they compete for those same bonding sites. So if I have something like an alcohol that's a polar solvent, it can come in and interfere with the bonding sites of the where the hydrogen bondings are taking place. So that's we use, for example, hand sanitizer. Hand sanitizer has alcohol in it, and so it disrupts the germs and things like that. pH change breaks down hydrogen bonding and disrupts salt bridges. And then organic solvents also disrupt salt bridges. And, if you have high concentration of ions, it does the same thing that the pH does, or changing the pH does. So it breaks down those salt bridges or disrupts those salt bridges.